Hello, everyone. Welcome to Foresight's Molecular Machines Group. Before we start, I just want to give you a brief announcement that we'll be meeting December 5th through 7th for our 39th, maybe it's not our 39th, but it's one of many Vision Weekends, our annual member gathering and conference. So if you like molecular machines, bio, nano, neuro, space, computation, we'd love to have you there. Well, without further ado, today we're so excited to have Dr. Samuel Thompson with us. They will be presenting on re-engineering protein engineering for organic solvents. So thank you so much.
Great. So I'm very excited to be presenting here, and I wanted to actually start off a little bit, considering where we are in politics right now, kind of honoring some of the institutions that have kind of led to this work and to what I'm doing. And so I grew up in West Texas, in a very small town of about 10,000 people. And when I was in high school, I applied to the Texas Academy of Mathematics and Science, which was a magnet program essentially for people from throughout the state. It's a public program that brought people together on a college campus and allowed them to simultaneously take their last 2 years of high school and their first 2 years of college at the same time. And that was the first opportunity that I had to do research in an academic setting then as a high school junior. And that really transformed my world, that one opportunity. And when I realized that science could take me to new places, science was the reason I was able to leave my hometown and go have this amazing experience in education, I really wanted to see how far that would take me. And honestly, it's taken me quite far. And so this is the kind of compendium of my academic background since starting at TAMS in 2006. But I want to focus mostly on some things that I was working on in Japan as a result of being an undergraduate at MIT and some of the opportunities that afforded. So I was in Osaka University doing a year-long research exchange stay. And I was working in the lab of Junichi Takagi. And while I was there, one of the professors started a conversation about projects that were abandoned, that things that had been popular in a field for a long time and abandoned. And one of the professors said, when I was in grad school, everyone wanted to put enzymes in organic solvents and replace all of organic chemistry with enzymes. And he said, it was just too hard. The enzymes die, and people just kind of gave up on it. And so that was really the moment that I told myself, like this moment in Japan, that this was the problem that I wanted to solve. So people wanted to use enzymes as organic catalysts because enzymes are generally the world's best catalyst in terms of their rate acceleration and their stereo and regiospecificity. The economy of steps, the energy efficiency, all of these things are very highly optimized through the course of evolution. And in the words of Joan Stubby, who was my biochemistry professor in my chemistry undergraduate, she used to talk about enzymes and say, eat your heart out, synthesis jocks. So it's a little bit ironic that I learned about the challenge of putting enzymes into organic solvents because many people would argue that research began in the MIT chemistry department. Alexander Klebanov is often regarded as the first person to study protein structure and activity in neat organic solvents, and many other labs contributed. When Frances Arnold started her lab, she was focused on directed evolution in aqueous organic co-solvent mixtures. And this work has been tremendously informative. It's still widely used in industry, but is also driven by what I would call a chemistry first perspective. You isolate a catalyst that you know and you care about, and you transfer it into your reaction. And this explains a great deal as to why this problem is so difficult and why there have been significant challenges in this area. You are forcing an enzyme that is optimized for water into organic solvents. And in response to that, there has been decades of research and a wide range of strategies that have been developed for stabilizing these enzymes in organic solvents because that transfer is thermodynamically unfavorable. Solubility is very low, and the solvent impairs stability and/or the activity in one of two general mechanisms. Polar and amphiphilic solvents generally denature the proteins because they can stabilize the unfolded polymer conformation of the protein, and non-polar hydrophobic solvents trap these fixed waters on the surface of the protein and rigidify it, and that loss of dynamics is concordant with a loss of the function of the enzyme. Now, again, I want to highlight that many of these approaches still have successful industrial application, but I think that there's a lot of functionality that we are missing out on. So I was in Japan in 2011 and 2012, and I thought about this challenge for about 8 years, going through the process of finishing my undergraduate and doing a PhD, because I wanted to be sure that I could bring something new to the table. And so at the end of that process, as someone graduating with a biophysics PhD with a background in structural biology and computational protein design, my first question when coming back to this problem was, what if you could build a protein from the ground up that thermodynamically prefers to fold and function in organic solvents? So rather than transferring the protein, it naturally partitions. And I'm going to call these proteins that don't exist in nature non-aqueous proteins. So if we understand the biophysical principles of how to design proteins specifically for non-aqueous environments, that unlocks quite a bit of technology that we can develop, in part because it's not just catalysts, but it's also materials and advanced polymer materials that are both atomically precise and infinitely scalable, to use the terminology of the Homeworld Collective. So first, we would expect to have very long-lived, stable industrial catalysts that are highly specific. And so, for example, we could produce biodiesel from the roughly 3 billion gallons of waste cooking oil that are produced in US restaurants. And if we can do that in an effective way, we could offset about 7% of US annual diesel consumption. We could effectively make all petrochemical and organic chemistry compostable, so greenifying many of the different processes that we need to use for modern synthesis. Second, we could develop technology that interfaces with natural hydrophobic environments, such as cytosolic lipid droplets, to develop novel therapeutics for viral infection, for obesity, for cancer proliferation. Third, we could use these self-assembling proteins to build self-healing membranes for flexible lithium-ion batteries, or nanoscaffold for 3D semiconductor chips. And finally, we can even think of, very broad, basic biology questions where we're imagining what would be the constraints on a biomolecule in a radically different planetary environment. So there's just a huge gamut of things that we can do when we have these templatable biopolymers that we can work with that don't function in water. So I think we're really, on the edge of quite a bit of downstream basic research and applied research. So as exciting as all that is, we can't work on that directly yet. What we need to know is, what do we have to do to unlock that huge potential within the non-aqueous protein space? And so I'm going to argue that what we have to do is we have to re-engineer our entire protein engineering ecosystem. And first, we need a demonstrated biophysical concept that allows us to use computational protein design to efficiently search through sequence space and predict the rare proteins that fold and function in non-aqueous solvents. We need expression platforms that allow us to produce enough protein for our assays and our characterization. We need the assays themselves that allow us to evaluate if the protein folds and functions in organic solvents. And then we need the modern tools of high-throughput screening for directed evolution. And finally, spectroscopy tools to determine the polymer solution, structure and solution, and also to evaluate if our designs match the computational model. So what I'm doing to build this ecosystem is combining computational protein design, molecular biology and biphasic reactions, NMR, and microfluidics. And so I'm going to talk through all of these stages of method development that we've been working on. And I'm incredibly lucky. I basically pitched this idea when I was applying to postdoctoral positions. I said, would you let me come work on this? And I was very lucky that people said yes. And I want to, give a huge amount of gratitude to Polly Fordyce here at Stanford and David Baker at the University of Washington, who gave me space to pursue this and opened up the very, very collaborative teams that they have at those institutes for me to work with. I'd also like to thank Kelly at the UCSF Marcor, some of whose data you're going to see later on in this talk, and then the people at the Stanford FACS core who've been helping me collect some of the data we have. These core facilities are real treasures of our universities, and we're so grateful to have them. And finally, I want to acknowledge fellowship funding from the Beckman Foundation and grant funding from the Homeworld Collective. Both of these groups have been exceedingly generous and just like wonderful champions of the project. And I want to particularly shout out the Homeworld Collective here, who've just been a great advocate for me and for the project. And their introduction to the Foresight Institute is part of the reason why I have this opportunity to speak to all of you. So I'm very grateful to Paul and Daniel and Paul and all the other people at Homeworld. So to start off, I want to talk about the proof of concept that we want to accomplish with this project, and specifically focused on designing proteins for water immiscible solvents. And the use of a water immiscible solvent allows me to work in biphasic reactions. And that use of two solvent phases allows me to sequester the molecular biology and the biochemistry and all the recent advancements that we've made in protein engineering in the aqueous phase from the octanol phase, where we really want to see these proteins fold and function for our applications. So what I envision is that I encode the design sequence in DNA, I express it in an in vitro reaction in this biphasic condition, and then just let proteins partition into the water immiscible non-aqueous phase and fold there, and there we can purify them and characterize them. For this proof of concept, we wanted to focus it on a very specific goal to demonstrate what we can do. And so I'm going to go back to the production of biofuels as just one example of one of the things that we could do with this technology, in part because it's important, but it's also something that's accessible and has a lot of lessons to teach us. So it's a great system because the transesterase activity is a model reaction for the production of biofuel, this ability to take one ester and cleave it with an alcohol. So this is, for example, how you might cleave the triacylglycerols in cooking oil and fatty acids to produce these biodiesel esters. And this is, I think, an untapped part of biofuel production. Most biofuel production has focused on the conversions of sugars and lignocellulases to alcohols, but we're not quite as effective at processing other parts of biomass material. And for example, the industrial process for this requires some pretty material-intensive acid-base catalysis. And there's a lot of cleanup involved in purifying the final fuel out of the process. And even just doing that acid-base chemistry introduces the possibility of side products that can foul your engine. So it's just never been a commercially viable process. And so the ultimate goal would be to take an involved non-aqueous catalyst, mix an anhydrous mixture of vegetable oil and an alcohol such that biodiesel esters are generated directly without any additional input, and that there's very little downstream workup in the process. But really, we want to understand, what does it even take to do this chemistry in the organic solvent itself? So to do that, we focused on very preliminary catalytic designs based off of helical bundles designed by Dec Wolfson's lab in Bristol. And in this work, back in 2016, they generated helical bundles that display a cysteine triad. And this is a classic motif that is conceptually very similar to the serine triads that you see in proteases. And so these catalytic triads activate the nucleophile within the cysteine so that this motif can cleave esters. And this is a great platform for kind of demonstrating a very simple system that can do the chemistry to convert fatty fats into biodiesels. This is just a scheme of the reaction here where the activated cysteine is able to cleave a reporter molecule at just a model substrate that has a strong absorbance into a carboxylic acid and the released fluorescent or absorbent moiety. Now, these catalytic helices are very rudimentary. They do not form a preordered active site like we see in many of the highly evolved enzymes. And so many of the rate enhancing features in natural enzymes are missing. It is just kind of a positioning of these residues next to each other. So we expect the reaction to be very slow with these. And in fact, that's what the initial publication observed. What they did is they mixed a 2-to-1 ratio of substrate to peptide. And in the first part of the reaction, you see this rapid release of the reporter molecule as the catalytic peptides react with the first half of the substrate in solution. But to react with the second step, the catalyst must regenerate itself. And so there has to be a solvolysis reaction, to process the next substrate. And this results in a much slower second phase of reaction. And this would be the true rate limiting step for catalysis. I should mention that this intermediate trapping is a very common problem for designed hydrolases and kind of one indication that we still don't fully understand a lot of the mechanisms of catalysis and how to generate good catalysts in proteins. But ultimately, what this system represents is it's a very simple, robust catalytic system with well-characterized kinetics. We know how it behaves, and it provides us with an accessible first goal. So we redesigned these catalytic helices with Rosetta by fixing the catalytic residues and adding some additional modifications to the simulation to accommodate organic solvents. The total peptide is about 30 amino acids, so we generated a fairly small design set of about 20,000 designs. We filtered those designs to find the most maximally different sequences and selected 24 total designs to test. So the next thing I needed to do was invent a way to actually test for peptides that thermodynamically prefer organic solvents. And I opted for a straightforward gel-based assay, considering the number of sequences that we had to test. So after expressing proteins in a biphasic in vitro transcription translation reaction with a layer of octanol on top, very similar to the concept I presented to you earlier, because they're in miscible phases, we can manually separate them. We can evaporate the solvents, and then we just resuspend everything in SDS buffer. But we add a flash labeling reagent to that. So when we ordered the genes for our designs to express them, we genetically encode this CCPGCC tag, which covalently binds to the flash 4,4, which is an arsenylated fluorescein molecule. And what this acts like is it's a small molecule version of GFP, essentially, that allows us to label the protein after it's been expressed and after it's been transported into whatever phases it is most thermodynamically favorable in. So we can fluorescently detect our proteins when we run them out on a page gel. So we want to check to see that our protein is detected in the non-aqueous phase, but then we also want to make sure that there's not contamination. So we run a silver stain on the gel afterwards to make sure that all of the biomolecules are appearing only in the aqueous phase. We don't want any water contamination in the non-aqueous phase. And that way we can determine that whether or not we have a true hit for our non-aqueous protein, if it passes both of these two tests. So here's an example of one of the hits that came from our screen. We have 3 replicate reactions. There we separated the biphasic mixture into the 2 soluble phases and then also the precipitate. And you can see here in the boxed bands that the fluorescent signal is in the octanol phase and in the insoluble pellet residue, but the signal is an order of magnitude lower in the aqueous phase. And you can also see from the silver stain gel that there are no biomolecules contaminating the octanol lanes. So we're confident that the fluorescence signal that we see in the octanol phase is from the partitioning itself. So now that we have a hit for a peptide that partitions into octanol, we need to be able to produce enough to characterize it. And the idea here is straightforward because these are very small peptides. We aimed to fuse them to a solubility tag and express them in E. coli. After expression, we use a genetically encoded cleavage site to release the hydrophobic peptide and extract it into octanol. And I want to give a shout out here to Gary Pielak for giving me the initial idea of how to do this, and Zhang Dai for giving me some suggestions that were really critical in troubleshooting this process. However, the troubleshooting of this process was quite massive because these constructs tend to express quite well and behave very poorly. We had several generations of this technology that were absolutely lethal to the E. coli as soon as we induced expression. But ultimately, I think we've gotten to a point that is workable. So here I'm showing you an expression gel. You can see that our fusion construct is the dominant band in the gel, indicating strong expression. And you can see that the cleavage is complete, causing that whole expression band to shift after the cleavage reaction. Obviously, we also want to purify the peptide from solution. So this is a purification from lysate. So because the peptide is so small and hydrophobic, it's actually not labeled by Kumasi and many of the other standard protein labeling methods. So I'm going to show you the fluorescence gel. And from the fluorescence intensity, we're estimating against a standard curve that we're getting about 5 milligrams per liter of total expression of our peptide, about 2 milligrams per liter of soluble expression. So on the low end of decent for heterologous expression, but also pretty good considering how much of the mass of the total construct is actually our peptide. And then the final product, purified product, is about 0.2 milligrams per liter of E. coli culture. So I think there's some optimization to be done there. I think there's some very obvious ways where we can improve that. But really, this has given us a brand new foothold for increased scale and economy in these experiments and has been an absolute game changer. So this expression platform is going to be critical for the structural biology, namely the NMR that I'm going to talk about in a few minutes, and we're really excited about that. So the next thing that we need an assay that can detect esterase activity in the octanol phase. And so again, quick reminder, we're looking for peptides that can perform this cleavage reaction where they convert triacylglycerol into separate biodiesel esters. What I'm going to model this as is the release of a fatty acid or a fatty acid ester from a fluorophore. And I want to use the fluorescence readout to detect activity that can only be attributed to a reaction that is occurring in the octanol phase. And this is a reaction that's more complicated than the typical enzyme reaction. And we're trying to do something kind of really novel and unexpected by demonstrating this in organic solvents. And so I'm going to walk kind of slowly through the logic of this experiment. So we started by identifying substrates that are excluded from the aqueous phase. And I took a common esterase substrate, this esterified 4-mu fluorophore. And this fluorophore is quenched until the ester is cleaved and the alkyl tail leaves. So I can modulate the length of this alkyl chain to see if we can sequester the substrate in the octanol. And in fact, when we measure the partitioning of substrates, we see that, again, these are both fairly hydrophobic, so the majority of them is in the octanol phase. But by modulating that alkyl tail and making it more hydrophobic, we can see that the partitioning coefficient is dependent on the length of the alkyl chain. And for this more hydrophobic 4-mu laurate, we can't even detect any of the molecule in the aqueous phase over the background signal. So we're fairly confident that's sequestered in the octanol. So the next thing I want to establish is some controls. I want to know that both of these substrates can be converted by an enzyme in an aqueous phase only. So we want to know that they're compatible with the enzyme. So we're going to mix our enzyme with our substrate and monitor the change in fluorescence activity. And when we have no octanol present, here we see that the background rate for substrate hydrolysis is very low. It's that gray line. That's our no enzyme control. And that we see a lipase in solution can convert both of those substrates. So there's no fundamental incompatibility between our enzymes and our two substrates. And I'm also going to point out on here the magnitude and the time scale. So we're seeing approximately 100 micromolar substrate converted in about an hour here. So now we want to ask what happens when we repeat this reaction in a biphasic condition. And in this case, we see reactivity with our less hydrophobic substrate. And for our more hydrophobic substrate, there's no significant signal over background, which is exactly what we are expecting. So compare to the aqueous-only control from the previous slide. There we saw approximately 100 micromolar substrate converted in an hour. Here, we're seeing roughly 10% of the total conversion in almost 40 hours for just the less hydrophobic substrate. And this is consistent, again, with what we expect, that the substrate is mostly secluded from the aqueous phase, but is able to occasionally transit into the aqueous phase. So it's a very slow reaction. For the more hydrophobic substrate, it's even slower. And effectively, on this time scale, we don't see any conversion of substrate. So the next thing we're going to do, and lastly, we're going to now keep the same substrate, keep this very hydrophobic substrate, and now change the catalysts and see if we can find catalysts that are locked into each of these two phases. And so here, again, just data from the previous slide, we see that the lipase does not convert our substrate. It's equivalent to the background signal. But when we add one of our hydrophobic peptides, we see a small amount of conversion of this substrate occurring during the course of reaction, and it is well above a background signal and our background error. Now, what I want to be very clear about here is I'm not claiming that this is catalysis. I'm not claiming this is an enzyme in organic solvents. What this is a substoichiometric reaction between a peptide and a substrate, but we're pretty confident that this is a demonstration that reaction is sequestered and specific to the water immiscible phase. So what we have is a method for detecting enzyme activity that is specific to the organic solvent phase, and that will allow us to do more screening and more catalyst optimization from here. So now that we have the basics of a fluorescence-based assay for a non-aqueous esterase worked out, I want to talk about how we're using microfluidics to achieve a CPU to GPU-like scale-up in characterizing these proteins. And specifically for this talk about characterizing non-proteins. And essentially, we want to go big by making our experiments very small. And much of the work that I've done in the area of droplet microfluidics has been done alongside Dr. Zijian Yang. So starting with Danny Toffick's work about 20 years ago, there's been a lot of interest in capitalizing on the robust FACS sorter technology that allows us to sort individual cells, but apply that to in vitro reactions. And that led to the development of double emulsion droplets or picoreactors that encapsulate picoliter volumes of cells or enzyme reactions within an oil shell. But because they have a structure that's a little bit like an artificial shell, it's an artificial cell, it's an oil shell that's suspended in an aqueous sheath, we can collect them at rates of approximately 10 to 1 million per hour per day, and then we can flow and analyze these reactions with commercial FACS sorters. So we generate these picoreactors using microfluidic devices like you see above. We can flow reagents into the chip to generate picoreactors at kilohertz frequencies. And we've demonstrated some improvements to this technology, particularly surfactants and sample loading. And that allows us to sort at rates of more than 1 million reactions per hour, which is the rate at that dashed line, and to do this continuously over multiple hours. So this means that we have an efficient, practical sorting process that can select over libraries of approximately 2 to 5 million in the course of a single day, or to perform more quantitative screening and characterization of about 100,000 sequences per day. And the optimized process looks like this. Here I'm showing you a mock selection where we can enrich for a rare fluorescent species that mimics a positive experimental results, the green fluorescent droplets. And we can do this without any obvious damage to the sorted picoreactors with both high purity and high recovery of our reactions. And so when I interviewed with Polly, I pitched the non-aqueous protein project to her because the lab had been working on these picoreactors, and I thought they were the ideal vehicle for screening proteins at massive scale in conditions that would kill a normal bacteria or other kind of biologic-based evolution platform. So by combining what I wanted to do in biphasic reactions with the high-throughput screening in droplets, that required making for the first time ever a triple emulsion where an octanol droplet sits in the core of the FACS-sortable double emulsions. And the ultimate goal is to use this triple emulsion that has an octanol core and these expression reagents and the activity reporter as a high-throughput screening platform for detecting catalysts that partition into octanol and are able to convert our fluorescent substrates. So in our recent publication, we demonstrated for the first time that it actually is physically possible to separate 3 immiscible phases in the droplet, or 3 different immiscible phases in the droplet, specifically this octanol water emulsion. Moreover, these triple emulsions are compatible with biochemistry, as we saw from our biphasic reactions. We can express GFP in these proteins, and they will survive for approximately 48 hours in solution, and then we can sort them and recover them by FACS. So to mimic the reaction that we wanted to do in terms of encapsulating fluorogenic esters that preferentially partitioned into octanol, we first demonstrated this using one of our more hydrophilic substrates that partitions slightly into the aqueous phase, and then we looked for turnover with a lipase that's present. So we were able to recapitulate using a bead-bound lysate the turnover of one of these substrates in our triple emulsion droplets, and we find that that conversion of substrate is dependent on having wild-type activity. So For example, when we mutate the catalytic residue in this lipase, we no longer see activity. So this is published work where we can detect over background the production of a lipase product in the presence of a aqueous octanol biphasic reaction in a droplet. But it's really only en route to what we want to do, which is combining what I was previously talking about and what we are working on with the double emulsion droplets for a true high-throughput screening platform. And that process is still in progress. And so I hope to be able to update you with that in the future. So I want to return now to the beginning of the process and examine how we can start to build more complex self-assembling structures and thinking about some of the applications around materials in terms of building scaffolds and membranes and 3D structures with these systems. So how do we go from a helix to something like that? And the obvious next step is doing the very basic biophysical work of examining how we can bring multiple helices together into controlled geometries. And this is really the inverse protein folding problem, but specifically for organic solvents. So what we need is a Rosetta for organic solvents, and eventually we're going to want to be able to develop something like an AlphaFold for organic solvents. So from a biochemistry and a biophysics standpoints, we often talk about protein folding in terms of hydrophobic collapse, where you have a nonpolar core that is sequestered from the environment and a polar surface on the protein that interacts well with the water. But from the perspective of polymer chemistry, protein folding is an intramolecular phase separation. We are separating a covalently bound solvent, essentially. We're a covalently bound biphasic environment within the peptide sequence itself. So if we want to create a protein that folds in organic solvents, we essentially just need to reverse those phases. We want both a very hydrophobic solvent on the outside, we want a non-polar surface to our polymer, and we want a buried polar core. The way that we can generate these proteins, because we have no prior examples, that means we can't use a lot of the recent machine learning algorithms for protein design. There really just is no training data for them. So we have to go back to classic Rosetta design, where we're using these biophysical score functions that incorporate van der Waals interactions, electrostatics, hydrogen bonds, etc., as well as some of the statistical potentials. So we represent our proteins as all atom structures with all nuclei being represented as a point, and we can sample both backbone torsion angles and side chain rotamers, and then we perform a simulated annealing where we're trying to find low energy structures using these methods. And so to turn this into a full design model, what we do is we first start with a backbone scaffold generated in Rosetta, so some assemblage of helices. And then we design a fully satisfied hydrogen bond network and bury it in the core of the protein. And then the rest of the protein and the surface is designed with hydrophobic amino acids. So the first libraries that we generated are based on helical bundles, a classic starting point for computationally designed proteins. And I generated sequences for 3-mer and 4-mer backbones with C3, C4, and D2 symmetries. For each of these designs, the monomer is just one single helix as before, no loops. And if we look at these designed proteins, we can see that there's several characteristics that I designed for. First, there's a fully satisfied hydrogen bond network in the core of the protein. Backbone hydrogen bond shields the polar amide groups from solvents. Proline caps at the termini protect exposed amides, and there are no loop regions with exposed amide backbone. And finally, we have a very hydrophobic surface with many large aromatic residues. And in total, we've done preliminary testing of about 500 genes using our gel-based assay. And we classified these proteins relative to the fluorescence signal for one of our model peptides. Greater than 50%, less than 50%, or not detected at all. And here I'm going to show that expression in the heat map as strong, weak, or none. And we can perform a similar analysis from the gel assay for partitioning. So we have the full preliminary results for our library, including some controls with transmembrane helices and designed proteins with C3, C4, and D2 symmetries. And in each of these design sets, we can identify proteins that express and a few that have strong expressions. And of these, we also found proteins that partition in each design set. We've been optimizing this screen to improve our hit rate, in part because one of the things that we've noticed is that improving expression is a clear optimization path forward. If we look at the data on aggregate, we see that about 40% of our transmembrane sequences can be expressed in vitro, but only about 16% of our designs express. But if we look at the designs that do express, we have a fairly significant hit rate. About 40% of the expressing proteins partition into octanol, and about 10% of those have what we classify as a strong partitioning. So we are optimistic that the more that we can produce these proteins, the more hits we're going to find. So now that we've identified some of our hits, we need to develop methods to structurally characterize them and evaluate our model. And with this, I've been working with Mark Kelly at UCSF. Now, I'm going to say, I hope you walk away from this project dissatisfied. I'm dissatisfied. I'm very happy with what we're doing, but there's so much more to be done. And I hope you're eager to hear the rest of the things that are in progress. This is part of the work that I'm going to be very upfront is very much in progress right now. So as a quick reminder, we are designing 3-mer and 4-mer helical bundles, and we have a few basic questions for each of those designs. We want to know, is it folded? Is it oligomerizing into a helical bundle? And how well does that structure match the original design structure? And for each of these questions, we have spectroscopy methods where we can expect pronounced and qualitatively different outputs based on the state of the protein. And each of these questions gets progressively harder and more expensive to answer. So we want to filter out failures early and often. And so to make the whole plan work, we're trying to demonstrate that we can collect these measurements, that we can collect samples that are good enough for these measurements, and really demonstrate the platform pipeline. So what we've been able to do so far is we've been able to demonstrate that we can collect high-quality CD and NMR spectra using unlabeled, chemically synthesized peptides. And I'm going to refer back to this when I answer some of the questions in the chat, so thank you for those. So here is the design model for one of our strong expression and partitioning hits from our screen, which is a tetramer of 33 amino acid monomers. We can run CD in butanol, which shows clear alpha helical signature for this peptide. And that signal is maintained up to 100 degrees Celsius. So these proteins are very stable in organic solvents. At least the secondary structure is very stable. We can also collect NMR spectra for the protein in pure methanol. And here I'm showing you a hydrogen NOSI experiment. For this spectra, the important features are the off-diagonal peaks. Those are for the weak transfer of magnetism between hydrogens through space over short distances. So the fact that we see cross peaks at all tells us that we have residues that are atoms that are in stable and defined spatial relationships. That kind of weak signal transfer does not occur if you have an unfolded protein. So we're very confident that these data together tells us that our protein adopts a stable fold. What we don't currently know is we don't know the actual solution structure, and that is what will require isotope labeling. So we're very excited with our recent developments in protein expression and purification that should allow us to actually produce those isotope labeled proteins. So just to quickly recap here at the end, what is the progress that we've made towards engineering a new protein engineering ecosystem for organic solvents? So I've shown you that we have computational design methods that allow us to enact our structural hypothesis for these proteins with a polar core and a hydrophobic surface. We have the ability to express these proteins and purify them in E. coli. We have the ability to express them using in vitro transcription translation, which allows a lot more high throughput methods. And these are both systems that we're optimizing, but they've been giving us a fantastic foothold. We have functional assays that allow us to detect the partitioning of these proteins in organic solvents, so we can detect what matches our thermodynamic hypothesis, and we're developing assays that actually allow us to look at catalytic activity that is specific to the organic solvent phase. We have the capacity to marry those assays with high-throughput screening, and we're very excited about the future potential for actually being able to do directed evolution by combining the 3 elements, the computational design, the expression, and the assay, into a FACS-sortable microfluidic droplet. And finally, we have a bit of a proof of concept that we can collect the data that we need to actually do structure characterization in solution in organic solvents for these proteins. So with that, again, I would like to thank my fantastic coworkers and collaborators, my funding sources from the Homeworld Collective and the Beckman Foundation, and then again, the Foresight Institute for inviting me to talk. And I am looking forward to your questions and addressing the ones that have already popped up in the chat.
Thank you so much. I know we have some questions in the chat. Maybe we address those first.
Yes. Okay, so I'm gonna start with Martin's question if I could.
Right.
So if the peptide is only 30 amino acids, why not synthesize the peptides by solid phase synthesis? Yes, this is absolutely where we started, and much of the data from the catalysis and the structure determination is using chemically synthesized peptides. We do want to move away from that for a couple of reasons. One is that it is, several $1,000s a pop. And so our ability to really vastly explore the sequence space and the structure space is limited there, but it is one of the best ways of getting pure peptide. We also have found that there are some contaminants in peptide synthesis that we're trying to move away from, and we're hoping that by producing these in-house ourselves in E. coli, we can get away from some of the impurities which seem to be intrinsic to the HPLC purification pipeline used in solid phase synthesis. And then the last thing is that, it's expensive enough to make these peptides by solid phase synthesis, but making isotope labeled samples for NMR is just financially intractable. We're talking about tens of thousands of dollars, to make a couple of different substitutions. So we'd be very limited in that aspect. So we really think that there's a reason why so much of protein biotechnology uses E. coli to make these proteins, and we want to capitalize on that technology as much as we can. The other thing about solid phase peptide synthesis is it's not yet high throughput over libraries of amino acids. Our ability to synthesize DNA and DNA libraries is much superior to our ability to synthesize libraries of peptides. And so we want to capitalize on that as well. Stephen asked, have you considered using persulfides as a superior nucleophile, or is the disulfide bond troublesome for your application? Oh, that's an interesting question. No, I haven't considered using persulfides in solutions. I guess that would require, at least as you have it described here in the chat, it would require an unnatural amino acid or maybe a conversion of the catalytic cysteine afterwards with some addition. I'm not much familiar with the use of persulfides in protein chemistry, but I would be very curious to follow up with this. I think it could potentially work. What I will note, for what it's worth, is I don't know that we see that particular approach being taken by natural enzymes. I don't know if that's something where enzymes in organic solvents could take advantage of something that is not frequently used in evolution, or if there's a reason why that might not be a good path for this particular system. But yeah, that's a very interesting suggestion that I follow up on.
I can suggest one thing and just look up the transsulfuration pathway, where the CBS gene protein switches from serine as a substrate, which is the alcohol amino acid, to cysteine as a sulfur amino acid, and it produces persulfides, and instead of, let's say, homocysteine.
Okay, I'm going to have to look at that much more carefully, but I think that's a very interesting suggestion that has never come across my radar before, and I will have to think about it very carefully. Thank you so much.
It is notable that the CBS enzyme is very prolific. It's very non-specific. It accepts
like some of these
many different substrates.
Some of these promiscuous kinases that have been used in various biotechnologies, where you just need to put a phosphate group on many different things. That sounds fantastic. Thank you. So Martin also asked, why didn't you choose beta structure as your model secondary structure? That is a fantastic question. And we have played around with beta structures. What we don't know kind of the full space of what proteins are compatible with organic solvents. And that is something that I would like to answer. We're a little bit limited in just the throughput of things that we can test. What I'll say is we had more early success with the helical designs. And I think that there's a couple of facts that can give us a little bit of intuition about this, but I also just want to state that this is so unknown that I can't say this with any particularly strong authority. This is just my own postulation on this. So we know from medicinal peptides that exposed amines and exposed amides are disfavorable to delivery of small molecules into cells. So it's hard to get through the membrane itself if you have a positive charge or if you have a nitrogen. So methylating your nitrogens is a very common process in medicinal chemistry. So we had this idea that we really needed to shield our nitrogens. And that is one of the things that is great about a helix, is that secondary structure almost kind of maximally shields the polar backbone from the solvent itself. And we also think that this is possibly why we see that these helices are so stable in organic solvents. I showed you that CD spectrum where you see alpha helical signature in our peptides up to 100 degrees Celsius. That's, very unusual for natural proteins and for proteins this small. And I think it could potentially be because there's just such a strong desolvation penalty. So I think it's possible to design beta structures, but beta structures in general, and loops definitely, tend to have more backbone exposed to solvent. And so I think it would just be a more complex design challenge to try to get a fully satisfied beta structure. And there's some things that we tried with that early on, and I think we just kind of had to simplify our first steps based on just the nature of the challenge.
One thing I'd observed is that small peptides that are designed to be in beta structure tend to be thixotropic, which would tend to mean that the hydrogen bonds are internal to the structure.
Yeah, it's really like the register. It's at the edges where like the sheets meet that you start to get more exposed backbone.
Yeah, that's why a lot of them go into various types of beta structure.
Yes.
Beta barrels and so on.
Correct, exactly. Lydia, I think that's the questions in the chat if you want to field any more from the group in general.
It looks like Tad has a question.
Yeah, thanks. This is a little bit too long to write in the chat. Thanks. This is an interesting talk. And I'm wondering, as you're thinking further along in your design process, when you mentioned designing from scratch to work in organic solvents, it got me wondering to what extent it would be useful to do either non-natural amino acids or even things other than proteins. And I was just wondering, is your main focus on proteins mainly just for practical reasons, like, well, we have E. coli that can make it, and we have computational tools for proteins, but not so much for sort of other molecules that might be potentially better suited for organic solvents?
Yes.
We just don't.
You've hit the nail on the head there. So where I come to this from is my high school research at TAMS was in material science, where we were making these functional hydrogels. And that's when I fell in love with proteins, because I was comparing the random polymerization in a lot of our material science, where you make these bulk polymers and you can really only control the structure at the micron scale, to what proteins can do, where you can control the position of individual atoms within the superstructure. So I think what I see myself is a material scientist who focuses on templated materials. And proteins are a wonderful example of templated materials. Ribosomes are, a great platform for how we can take something from a computer, encode it into one language, i.e. DNA, and then output multiple copies of our templated polymer, i.e. the protein, in a simple reaction. And all of that kind of scales and allows us to take advantage of high-throughput screening. And I really think that high-throughput screening is going to be necessary to understand the physics of this space, just because we don't have naturally existing data sets to work with. We're really going to need to be able to scale up to collect a lot of data. So that's why I'm interested in that. But I'm super interested in what ribosomes can do to make polymers other than proteins. So Hiroaki Suga, for example, has done incredible work at looking at alternative backbone chemistries that can be accommodated by ribosomes. And so I would really, in the future, like to be able to push that ability to make templated, defined polymers into non-peptide spaces.
Yeah, that makes sense. So, yeah. So thanks. Thank you.
Great. I think we have time for one more question, if anyone has any questions.
I did have a possible follow-up question relating to, instead of hydrogen bonds, using disulfide bonds as the secondary structure element in what you're trying to achieve, because hydrogen bonds maintain a certain water solubility, whereas disulfides are pretty hydrophobic.
That is true. Part of the reason that I have avoided that is that we use the disulfides in our labeling technology. And so I've tried to avoid having extra disulfides around. There have been some, so I guess it was Benjamin Basanta in David Baker's lab, I think maybe 2013, tried to take a designed protein and kind of do this process where they put a lot of disulfides in the core of the protein and made the protein very hydrophobic on the surface. They were never able to get those proteins to express so that they could characterize them. So that study stopped at the computational level, but it is something that I have been thinking about. I guess I'm interested in anything that will allow us to stitch helices together. I've thought a little bit about, even kind of click reactions to bring multiple different secondary structure elements together into a tertiary structure. So I'm open to any possibility that works.
Okay. Well, that brings us pretty much to time. So thank you, Samuel, for coming on.
And thank you for having me. This has been delightful.
Yeah, it was great to have you here and hear about your work. And if there are any follow-up questions, I'm wondering if people could email you.
Yes, so you can find me, a Google search, Samuel Thompson at Stanford University. You can find me on LinkedIn. Usually I'm pretty Googleable, just with my name and Stanford.
Great. Well, hope everyone has a great rest of your day and see you next time.